Welcome to Part 2.2 of Excipients for Oral Tablet Formulations. This section focuses on three key fillers and their roles in tablet formulation. In this part, we'll explore the key characteristics of lactose, mannitol, and dibasic calcium phosphate. We'll also examine how their flow, compressibility, and stability behaviors influence formulation design, followed by a brief summary and transition to the next section. Next, we move to another widely used inorganic diluent, dibasic calcium phosphate, commonly known as DCP. It's a cost-effective, stable, and free-flowing excipient frequently used in both pharmaceutical and nutraceutical tablet formulations. Dibasic calcium phosphate, or DCP, is available in two primary forms, anhydrous and dihydrate, each with distinct processing characteristics. The anhydrous form has higher porosity and superior compressibility, making it particularly well suited for direct compression and enabling fast tablet disintegration when properly formulated. The dihydrate form, on the other hand, exhibits better handling during wet granulation and consists of robust, clean processing particles ideal for granule formation. Choosing between the anhydrous and dihydrate forms depends largely on the manufacturing method and the moisture sensitivity of the active ingredient. Dibasic calcium phosphate is widely chosen by formulators for several practical and performance-related reasons. It serves as an additional source of calcium, making it highly suitable for nutraceutical and over-the-counter supplement tablets. Despite its brittle nature, DCP provides good compaction properties, since its fragmentation during compression creates new bonding surfaces that enhance tablet strength. It also demonstrates excellent flowability, ensuring uniform dye filling even at high tableting speeds, an advantage in large-scale production. And finally, DCP is cost-effective compared with many organic fillers, offering both performance and economic benefits in formulation design. Dibasic calcium phosphate is an insoluble, non-swelling excipient. Because it does not generate sufficient internal stress during hydration, it cannot independently promote tablet breakup after compression. To ensure proper disintegration and effective drug release, a swelling-type superdisintegrant must be incorporated, such as cross-carmylose sodium, sodium starch glycolate, or crosspovidone. The concentration and placement of the disintegrant, both intra and extragranular, should be carefully optimized to achieve rapid and complete tablet disintegration. In short, while DCP provides excellent strength and flow, its lack of swelling makes pairing with an efficient disintegrant essential for performance. Dibasic calcium phosphate exhibits a mildly alkaline pH, typically around 6.5 to 7 in aqueous dispersion. While this can be advantageous for neutral or basic drug substances, it may pose challenges for acid-sensitive active ingredients, which could undergo degradation or discoloration under basic conditions. Furthermore, for drugs whose solubility decreases at higher pH, the use of DCP might reduce bioavailability. Therefore, when formulating with DCP, compatibility and stability studies are essential. If necessary, buffering agents or pH-modifying excipients can be employed to maintain drug stability and performance. Because of its brittle crystalline nature, dibasic calcium phosphate can be abrasive to tablet punches and dyes during prolonged production runs. This effect becomes more pronounced under high compression forces, where repeated fragmentation of DCP particles increases wear on metal surfaces. To minimize abrasion, several strategies can be employed, using polished or coated tooling such as titanium nitride or chromium coatings, applying adequate lubrication with magnesium stearate or stearic acid, and implementing regular inspection and maintenance of compression equipment. These measures ensure both tooling longevity and consistent tablet quality throughout production. Unlike plastically deforming excipients such as microcrystalline cellulose, dibasic calcium phosphate is largely insensitive to lubricant concentration. Its brittle fracture mechanism allows consistent tablet hardness and tensile strength even at higher levels of magnesium stearate. This property offers greater flexibility during formulation, making DCP especially useful when stable compaction performance and minimal weight variation are required, even under high-speed tableting conditions. Let's move on to lactose, one of the most common and versatile diluents used in tablet formulation. Lactose, a disaccharide of glucose and galactose, exists in several solid forms, each with distinct physical and functional properties. The main types include A-lactose monohydrate, anhydrous A-lactose, anhydrous V-lactose, and amorphous lactose. These forms differ in crystallinity, solubility, and moisture behavior, which influence their suitability for wet granulation or direct compression. Commercial lactose products often combine two or more forms to achieve balanced flow, compressibility, and dissolution characteristics for specific manufacturing processes. A-lactose monohydrate is the most widely used form of lactose in tablet formulation, particularly suited for the wet granulation process. It contains about 5% water of crystallization, which is structurally bound within the crystal lattice and remains non-reactive. This bound water does not participate in chemical reactions or promote degradation of moisture-sensitive actives, ensuring stable performance under normal processing conditions. A-lactose monohydrate exhibits excellent flow and compressibility after granulation, producing tablets with consistent hardness and low friability. Its stable crystalline structure ensures good storage stability and reliable performance during scale-up, making it a dependable choice for robust and reproducible tablet manufacturing. Lactose can chemically react with primary or secondary amine groups present in certain drug molecules. This reaction, known as the Maillard reaction, results in browning or discoloration of the formulation and may reduce drug potency. It occurs when the reducing sugar portion of lactose reacts with amines under heat or humidity, forming brown-colored complexes and degradation products. Therefore, lactose should be avoided in formulations containing amine-based active ingredients, such as amphetamine, fluoxetine, or sertraline. If lactose must be used, selecting anhydrous or low reactivity grades and maintaining controlled humidity and temperature during storage can minimize the risk of this undesirable reaction. Agglomerated or granulated alpha-lactose monohydrate is frequently used in direct compression formulations. These processed forms provide improved flowability and compressibility compared to pure crystalline lactose. Anhydrous lactose, on the other hand, is specifically developed for direct compression and delivers consistent tablet strength without granulation. It's especially preferred for moisture-sensitive active ingredients since it contains no water of crystallization. Lactose is a brittle material that undergoes fragmentation rather than plastic deformation during compression. This characteristic allows anhydrous lactose to retain compactability even after reprocessing or handling. Its high fragmentation tendency creates fresh bonding surfaces, enhancing interparticle adhesion and tablet strength. Anhydrous lactose exists in two crystalline isomeric forms, alpha and beta, with the beta form promoting faster disintegration and higher solubility. Spray-dried lactose is a co-processed mixture of crystalline alpha-lactose monohydrate and a small amount of amorphous lactose. The crystalline portion provides excellent flow, while the amorphous phase enhances binding and compressibility, making it ideal for direct compression. However, spray-dried lactose is sensitive to moisture. Humidity can trigger recrystallization, leading to reduced tablet strength and higher friability. It should therefore be stored under low humidity conditions. Overall, it offers a balanced combination of flow and compactability, making it one of the most widely used lactose grades in direct compression. Next, we'll discuss mannitol, another key diluent widely used for both pharmaceutical and nutraceutical formulations. Mannitol is a highly stable, non-hygroscopic diluent valued for its chemical inertness and compatibility with most active ingredients. It provides a pleasant cooling sensation and smooth mouthfeel, making it the excipient of choice for chewable and orally disintegrating tablets. Mannitol exists in several polymorphic forms, each exhibiting distinct flow and compression properties suitable for either direct compression or wet granulation. Overall, its excellent stability, pleasant sensory properties, and broad compatibility make mannitol a versatile and patient-friendly excipient. Mannitol is available in several physical grades, each designed for specific manufacturing methods and performance requirements. Crystalline grades are typically used in wet granulation, where they provide structural integrity and low hygroscopicity. However, pure crystalline mannitol has limited flow and binding capacity, restricting its use in direct compression. To overcome this, spray-dried and granulated grades have been developed. These forms feature spherical particles with improved flowability and compressibility, making them ideal for chewable and orally disintegrating tablets. Mannitol offers excellent chemical stability and does not participate in Maillard-type reactions with amines, though its brittle fracture under high pressure may reduce compactability. Higher lubricant levels, such as magnesium stearate, help maintain smooth ejection without sacrificing strength. Overall, mannitol's grades provide flexibility across different manufacturing techniques, combining excellent flow, pleasant mouthfeel, and low moisture sensitivity, making it one of the most versatile excipients in solid dosage formulation. Now let's compare the performance characteristics of the major diluents and fillers used in oral solid dosage formulations. When assessing diluents and fillers, the key is how their physical and structural traits translate into tablet performance. Crystallinity and polymorphism dictate compressibility. Highly crystalline materials like DCP offer excellent flow, but limited bonding, while amorphous or spray-dried forms, such as lactose, deform better and yield stronger tablets. Particle size and distribution influence uniformity. Narrow, consistent ranges improve mixing, dye filling, and weight control. Finally, particle shape and surface texture affect packing and bonding. Irregular shapes enhance interlocking and strength but reduce flow, requiring a careful balance between flowability, compactability, and mechanical stability. In formulation science, a diluent's performance depends not only on its chemistry, but also on how it interacts with moisture, density, and mechanical behavior during processing. A controlled level of moisture can enhance compressibility, but excess leads to soft tablets and sticking, while dryness causes static and poor bonding. Density influences dye filling and tablet size. High-density powders improve dose efficiency, whereas low-density ones aid disintegration but may cause flow issues. Solubility and wettability govern dissolution, especially in direct compression systems, where poor wetting can limit drug release. Ultimately, the balance between plastic and brittle deformation defines compression behavior. Blends such as MCC with lactose achieve optimal rearrangement and bonding, producing robust, high-quality tablets. Choosing the right excipient is therefore critical. It determines not just whether a formulation compresses, but whether it performs. In this section, we explored how diluents and fillers affect the manufacturability and performance of oral solid dosage forms. We reviewed key excipients, microcrystalline cellulose, lactose, mannitol, and dibasic calcium phosphate, and discussed how their properties influence tablet quality and stability. In Part 3, We'll move on to the next excipient group: binders, disintegrants, lubricants, and glidants, and see how they enhance tablet strength, disintegration, and manufacturability.